Hi, I'm Robin Shrestha. I'm going to talk on this paper which focuses on cloning of a quantitative resistance gene. LR34 is a gene which confers a resistance to three rust species, that is leaf rust, stripe rust, and stem rust, and one powdery mildew species. This gene is also associated with development of a leaf tip necrosis. Interesting thing about LR34 gene is that it is a durable resistance gene and it has been used for over 50 years without being overcome by different rust species. Here in the first picture, we can clearly see the rust resistance line in the right-hand side and the rust susceptible line in the left-hand side. In the second figure, we can compare the area of leaf infested by rust in the three successive flag leaves on resistant line on left-hand side and susceptible line on right-hand side. And the third picture depicts the leaf tip necrosis on the line with LR34 and without leaf tip necrosis on the line without LR34. The LR34 gene is located on short arm of the chromosome 7D. The research group on this paper were able to reduce the target interval to 0.15 centimorgan. They did it through a map-based cloning approach and 3 high-resolution parental accession. When LR34 containing wheat variety Chinese spring was fully sequenced, 8 candidate genes were found, 1 hexose carrier, 1 ABC transporter, 2 cytochromes P40, 2 lectin receptor kinases, 1 cysteine proteinase, and 1 glycosyl transferase. Since cysteine proteinase and the glycosyl transferase were interrupted by repetitive elements, they were excluded as candidates for LR34. However, one of the 2 receptor kinases, the hexose carrier and ABC transporter, were co-segregating with LR34. To determine if one of those co-segregating genes corresponds to LR34, 3 analyses were conducted. First, sequence differences in the coding regions of the 3 pairs of plus minus LR34 parental lines were examined. Here, a consistency in a sequence polymorphism was found between the alleles of all 3 parental pairs present in the putative ABC transporter gene. Second, locus-specific DNA fragments of the 6 candidate genes were sequenced on the 2 gamma radiation and 6 sodium azide LR34 mutants selected for loss of function of the LR34 resistance. Here, each mutant showed sequence alterations in the putative ABC transporter gene. Third, to test for the presence of additional mutations in the other sequence co-segregating with LR34, DNA fragments of other 5 candidate genes and intergenic regions on the 6 mutants were analyzed. Here, sequence polymorphism was not found. Thus, these studies suggest that the 8 independent mutations in the putative ABC transporter gene are not only because of the high mutation frequency and can be concluded that LR34 is a durable resistance gene. Furthermore, LR34 also confers resistance against stem rust at sealing stage at low temperature. As in the figure, we can see that at 31 days post-inoculation of the pathogen, the infected colonized area is bigger in mutants compared to wild type. Similarly, the leaf rust sporulation was seen in mutants earlier than in the wild type. In the wild type, it was visible at 45 dpi, and in mutants, it was seen as 34 dpi. LR34 is present in susceptible plants too, but the LR34 allele present in resistant plants encode ABC transporter that has 1 point mutation in an intron that might affect gene splicing, lacks the amino acid phenylalanine, and contains 1 different amino acid, that is tyrosine, which is replaced by histidine. These polymorphisms might have changed the specificity of the ABC transporter so that it secretes more toxic compounds or higher levels of the same toxic compounds to combat against pathogens. Regarding the source of origin of LR34, 3 breeding lineages of wheat germplasm have been found. They are Far Eastern germplasm, spring wheat lines from the North and South America, and winter wheat from Europe. Here, A and T nucleotide at intron 4, 3 base pair deletion at exon 11, and C and T nucleotide at exon 12 were distinguishing features for presence absence of LR34, which were same in all 3 breeding lineages. The same resistant haplotype found in these 3 breeding lineages suggest a single origin of LR34. To determine if the Hence, in resistance between seedlings and adult plants in the lines containing LR34, reverse transcriptase polymerase chain reaction was performed using uninfected leaf material of near-isogenic lines, Thatcher and Thatcher LR34. LR34 was expressed very low in seedling, whereas it is expressed higher levels in older leaves. On the other hand, there was no visible difference in expression between resistance and susceptible plants. Furthermore, LR34 gene is expressed at higher levels in older leaves, especially the flag leaf. Presence of LR34 resistance gene caused leaf tip necrosis. Northern blot analysis revealed that SVS40, which is known to be upregulated during leaf senescence, was highly expressed in uninfected flag leaf tips of Thatcher LR34, but not in Thatcher and the 6 azide-induced mutants. This suggests that LR34 stimulates senescence-like processes in flag leaves. All of these studies confirms that LR34 gene is a pleiotropic and provides broad spectrum resistance to leaf rust, stipe rust, stem rust, powdery mildew, and also controls the phenotypic trait LT1, which represents for leaf tip necrosis.